Hello students, today I am Dr. Rahul Kaushik. Today we are going to cover another topic of your syllabus. Today we are going to cover the production techniques and the estimation and utilization pattern of phytoconstituent digoxin. So digoxin, if you can see in the slide that digoxin, as the name itself suggests, it is something obtained from Digitalis. If you have learned the if you have studied the drug Digitalis. So if you look at the structure of this digoxin, you can see there are two parts. This is sugar part and this is non-sugar part. So like anthraquinone glycoside, we have this glycoside which is having a sugar part and a non-sugar part attached by oxygen, and this is the cyclopentane of phenanthrene ring, where the OH, this is the 12th position, the molecule is hydroxylated at 12th position. So if you see the introduction, the digoxin, it belongs to cardiac glycosides. The cardiac glycosides, they are those glycosides which are having their effect on the cardiac muscles, especially the cardenolites C23. This digoxin, it belongs to the C23 group of cardiac glycosides. The drug digoxin, it is isolated from the foxglove leaves. Foxglove leaves, this is the common name. If you look at the botanical name of foxglove leaves, it is Digitalis purpurea and Digitalis lanata. The drug belongs to the family Scrophulariaceae. It is very important to mention here that since the word foxglove leaves is used, it is have nothing to do with the fox or the gloves of the fox. So it is the name given anonymously to this plant. So it is it have no resemblance with the gloves of the fox. So this digoxin, it is a cardiac glycoside. It belongs to the C23 group of glycoside. It is obtained from the leaves of Digitalis purpurea and Digitalis lanata, and the plant belongs to the family Scrophulariaceae. Cardiac glycosides, which is a steroidal saponin, and the digoxin, it is hydroxylated at C12. It is a white color crystalline powder and it is odorless. So this is the introduction of digoxin. If you look at the plant, beautiful plant digoxin, these are the this is the terminal position. These flowers, they are present at the top or the terminal location of your plant. This is the plant with broad leaves, and these the flowerets, it is a compound flower which compose of various florets. And if you turn the flowers of Digitalis opposite, or you can say if you opposite the leaves, if you can opposite the flowers of these foxglove, it looks like the bell shaped. So it is a biennial herb which is grown in England, but due to its medicinal value, it is also cultivated in USA, Europe and India also. Solubility, it is soluble in ethanol, but it is insoluble in water. So solubility matters because we are going to isolate, we are going to extract the phytoconstituent from the parent drug or from the parent plant. Use of these cardiac glycoside, as the name suggests, it is it acts on the cardiac muscles, or they are having cardioprotective action. So cardiac glycosides, digoxin, and it is insoluble in water. If you look at the cultivation processing of Digitalis, this is the complete botanical parameters of your foxglove leaf. These are the leaves, foxglove leaves, from which you can obtain the digoxin. These are the terminal flowers of your Digitalis leaves or Digitalis plant, and you can also see the beautiful flowers, multicolored beautiful flowers of Digitalis plant. The flowers, they have the very low or very minute concentration of your digoxin, but the main source for isolation of digoxin are the leaves of the Digitalis plant. This is the powder of the Digitalis leaves, from which we are going to isolate our phytoconstituent digoxin. So, let us move on to the isolation part. Before moving on to the isolation part, we should know how this phytoconstituent is going to act on your body, because we have seen the use of this cardiac glycoside on the cardiac muscles. So, its main mechanism of action is on the cardiac muscles. If you look at the mechanism of action of on the cardiac muscle, there is a normal mechanism in which we have outlet or exit of 3 sodium in exchange of 2 potassium ions. 3 sodium ion they go out of the muscle cell. This is the muscle cell, this is the cardiac muscle cell, which is called as cardiac myocyte. There is exit of 3 sodiums in exchange of 2 potassium through ATPase. This is the channel, and simultaneously there is another channel from which there is an exit of 3 sodium outside and inlet of 2 calcium. So, what happen when you take the digoxin? It enters your body, it reaches to the your cardiac muscles. This Digitalis or this digoxin, it inhibits the sodium pump. This pump was inhibited by the action of digoxin, and due to the inhibition of this pump, sodium is unable to go out, and potassium is unable to come in. So, what happen? More of sodium they get accumulated inside the cell, more of sodium. Because digoxin, it blocks the ATPase, sodium ATPase pump, but it has no effect on this calcium pump. So, what happen? Excess of sodium, it goes out through this channel, and more of calcium, it enters inside the cell. More calcium enters inside the cell in exchange of more sodium, because there is no potassium sodium exchange. So, what happen? This increased calcium concentration in your cardiac muscle cell, it causes the fiber shortening of the cardiac muscle fiber. So, you can see the cardiac muscle fiber. So, it causes the excess of calcium, it causes the shortening of fiber. So, this length it changes to this. Okay. In this way, your failing heart, the decreased activity of your heart due to the excess of calcium in your body, in your cardiac muscle, it causes the increased contraction, and the ventricular output or the ventricular performance increases due to the excess of calcium or due to the excess of contraction of your ventricles. So, in this way the declining or failing activity of your heart, it gets improved when you take the digoxin. So, it goes to the normal. So, this is the mechanism of action by which digoxin is going to act on your body. Production technique, production of digoxin, the method for production of digoxin is very simple, and here we obtain the drug in the form of a mixture. The moistened foxglove leaves, the leaves of Digitalis purpurea or Digitalis lanata, they are moistened, and an enzyme is added to the mixture. The enzyme glucosidase was added, and a temperature of 37 degrees Celsius was maintained with the moistened leaf, and there takes place the digestion of the glycoside, and the excess of glucose, it is removed from the extract, and the content is extracted using ethanol. After deglucosing, after the removal of excess of glucose, the content was extracted using ethanol. After extracting the content with ethanol, the ethanolic extract is then treated with lead acetate. Lead acetate treatment was done to remove excess of tannin, or you can say the lead acetate treatment or the lead acetate was added to remove the tannins and other phenolic compound from the ethanolic extract which we have obtained in the last stage. The filtrate, after removing the excess of tannins and other phenolic compounds, is then evaporated to dryness using your rotary vacuum evaporator. The residue is then treated with 90% alcohol and filtered, and the solvent is again evaporated. So, this is a process of evaporation, again mixing with the ethanol or the solvent, and again it is filtering and then solvent removal or solvent evaporation using rotary vacuum evaporator, and after that you can obtain the white crystalline powder of digoxin. So, this is the short method and it is the quickest method for isolation of digoxin from the Digitalis leaves. When you have isolated the Digitalis leaves, you should confirm that whether the isolated component contains the basic nucleus of cardiac glycoside or the steroidal saponin glycosides. So, for that you have a specific test where what we do, we took the sample and treat the sample with acetic anhydride, and the mixture is then added with a few drops of concentrated sulfuric acid. After addition of concentrated sulfuric acid, the initial reddish violet color, this reddish violet color, it was obtained by addition of acetic anhydride and sample. But as soon as you add the concentrated H2SO4, the reddish violet color, it gets converted into green color, and this test is called as Lieberman-Burkhard test or acetic anhydride test. This test detects the cholesterol nucleus. So, this test is very common for detection of cholesterol or steroidal nucleus. And now you have isolated the drug, you have confirmed the presence of cholesterol nucleus inside the extract or your isolated phytoconstituent. Moving on to the some instruments which we have used for carrying out the extraction, evaporation and separation of phytoconstituent from one solvent to another. So, this is the Soxhlet apparatus. It consists of 3 parts: the heating source, the extractor and the condenser. So, what we do, we took the solvent here, we place the drug here, the solvent vapors, they get evaporated by application of heat, solvent evaporate and it enters the extractor through this side tube. As soon as it enters the side tube, it enters the extractor, it tries to escape from the top, but at the top you are having the condenser. Due to condenser, the solvent vapors, they get condensed and they fall on the drug. So, here the drug and the solvent, they came in contact with each other, and in this way the extraction carries out. And as soon as this extractor fills up to this part, a siphoning cycle takes place automatically due to the pressure variations, and during this siphoning cycle, the content which is placed with the content or the extract of drug with the solvent, it moves down again to the main solvent chamber, and this is called, this one cycle is called as siphoning cycle. And in this way we carry out 3, 4, 5, 6, 7, 8 siphoning cycles for carrying out the Soxhlet extraction, and the siphoning the time for one siphoning cycle depends upon the drug, and it depends upon the solvent being used for carrying out the extraction of your drug. This is the separating funnel. We have already mentioned the use of or the functioning of this separating funnel in our previous lectures. So, what happen here in this separating funnel, we can partition a phytoconstituent from a low soluble solvent to a more soluble solvent. Suppose we are having a solvent A in which we are having our phytoconstituent X, and we want to transfer the phytoconstituent X into another solvent for further processing. So, for that you can use a solvent you can use a solvent which is having more solubility for X than the A, and when you use that particular solvent B for extraction, and you will put both the solvent A and B in this separating funnel, and you should shake the mixture, and you can see the maximum content of the phytoconstituent X, it gets partitioned into the solvent B. So in this way you can use the separating funnel. Normally the separating funnel can be used for separation of two immiscible liquid, but in phytochemistry you can also use this particular instrument or this particular vessel or instrument for partitioning your phytoconstituent from one solvent to the another. This is the rotary vacuum evaporator. This we have also discussed the use of this rotary vacuum evaporator. This is the round bottom flask. The extract which we have to evaporate was put in this extract along with the solvent. We want to evaporate the excess solvent from this extract. So this instrument is provided with the mechanism of rotation. This rotates. As soon as you provide the heat through this water bath, the extract, the solvent molecule in this extract, they form a fine thin layer over here, and because it is rotating continuously, so the molecules from that fine film of solvent, it gets evaporated very quickly at a very low temperature, and simultaneously we have also provided with the negative pressure using vacuum, because we have used a vacuum. So this vacuum tube, it creates the negative pressure, and due to that negative pressure, the solvent which is evaporated normally at 80 or 90 degrees Celsius, it starts to evaporate at a very low temperature of up to 40 to 50 degrees Celsius. So what we achieve in this assembly, we have recovered the solvent. When the solvent vapors they evaporates from this film, the solvent, due to the negative pressure and pump, it suck down here. Here the solvent vapors, it get condensed. After condensing, the solvent gets collected in this collector. So what we have achieved here, we have isolated the solvent, and we have prevented the thermal degradation of our phytoconstituent. So this technique is very important for phytochemistry or for phytochemists, because we can reuse this recovered solvent, we can reuse this recovered solvent for further extraction procedures of the same drug. So in this way you can minimize the consumption of solvent and also preserve the thermal integrity of your phytoconstituent. So this is the rotary vacuum evaporator we have used in this extraction procedure. Then let us discuss the properties of digoxin. So you can see in the picture, this is the crude digoxin or the leaves of digoxin, Digitalis purpurea, and this is the purified crystals of your digoxin. You can see the melting point of digoxin is around 248 degrees Celsius, and the boiling point of digoxin is around 661.93 degrees Celsius at 760 mmHg. So this is the pressure at which the boiling point is around 661.93 degree Celsius. If we talk about the solubility of digoxin, it is insoluble in water, as we have seen in the very first slide that it is insoluble in water, but it is very soluble in ethanol. The density of digoxin is more than water, it is 1.4. Storage, if you want to store the digoxin at in the purified form, you can use refrigerator also, because it is having a practically possible storage temperature that can be achieved through the use of refrigerator. It is available or it occurs in the form of white or white crystalline solid powder. Water solubility of digoxin is very low. If we if we look at the water solubility at room temperature of digoxin, it is around 46.08 mg per liter. It means that in 1 liter of water only 46.08 mg of digoxin is soluble. If you look at the lambda max of this digoxin, lambda max, here we have two lambda maxes, 255 and 251 nanometer lambda max. Lambda max is the absorbance maximum. This is the wavelength at which this particular phytoconstituent digoxin, it shows the maximum absorbance in the UV spectrophotometer or UV radiations. Molecular weight of your phytoconstituent digoxin is around 780.94. So, this is the molecular weight of your digoxin. Moving on to the estimation part, suppose we have isolated the compound, we have tested the compound for its identity, and now we have to estimate or we have to quantify or qualify qualitative analysis or quantitative analysis of digoxin in our drug sample. So, we can use the techniques like HPTLC and TLC and HPLC. So, there is a difference between HPLC and HPTLC. HPTLC is high performance thin layer chromatography. It is the version or the advanced version of thin layer chromatography, whereas the HPLC is high pressure liquid chromatography. So, in this way you can estimate or you can analyze your phytoconstituent in your drug sample. So, if we took the if we take the HPTLC and TLC, here we have used the silica gel, pre-coated silica gel plates which are available in the market. We have used the pre-coated silica gel plates. The mobile phase for TLC analysis we have used is ethyl acetate and methanol and water in the ratio of 81, 11 and 8. 81 part of ethyl acetate, 11 part of methanol and 8 part of water. It is very important to look here that because the phytoconstituent digoxin, it is insoluble in water. So, we have used a very small quantity of water in the mobile phase. So, through these mobile phases you can also analyze the solubility pattern of your phytoconstituent. Flow rate or injection volume is not applicable in TLC. Moving on to the detection of your developed TLC plate. If you have developed the TLC, you got the chromatogram. In case you are unable to visualize them through your naked eyes, so for that you can utilize the derivatization technique for the visualization of your phytoconstituent on the TLC. So, for that you can use Cadez reagent, you can spray the Cadez reagent, heat the plate, and you can observe the plate at under UV radiations at long wavelength ultraviolet radiations 366 nanometer. So, you can see the RF value of digoxin, you can see the RF value, you can observe the RF value of digoxin at 0.65 on the TLC plate of your developed plate. The sample for this analysis was prepared by dissolving 250 mg of drug, your Digitalis drug or Digitalis leaves, which is added with the which is extracted with the 20 ml or 25 ml of 50% methanol. The mixture is then ultrasonicated. The mixture of the drug which are the leaves of your Digitalis purpurea or Digitalis lanata, and the so extracting solvent which is methanol, 50% methanol. This mixture is then ultrasonicated. Ultrasonication was done in sonicator for 1.5 hours for 1.5 hour in ice water bath. Normally in sonicator we use high temperatures for extraction, but here we have used ice water, or you can say the cold water for extraction of your digoxin from the drug. After extraction through ultrasonication, the mixture is then filtered. The filtered mixture is added with 3 ml of methanol and 50 ml of ethyl acetate, and this mixture, this entire volume is then used for sampling or for placing sample on the TLC plate. The standard for this analysis, TLC analysis, was prepared by dissolving 1 mg of standard digoxin. Standard digoxin is available in the market. You can obtain it from the Merck or the Sigma. You can take the 1 mg of that standard and dissolve it 1 ml of methanol and use appropriate volume of that particular standard and the sample for application on the TLC plate. So, this is the TLC analysis of your phytoconstituent digoxin. If you look at the HPLC analysis, in HPLC analysis we have used the we have used the column Cosmosil. Cosmosil is the brand name. Cosmosil is the brand name of column which we have used, and it is C18 column. C18 column means the column which we have used is having the stationary phase which is coated with the C18 polymer. So, in this way it provides a non-polar stationary phase for the polar solvents as mobile phase. So, these are the specifications for column. The pore size for this stationary phase is around 5 micrometer. The mobile phase for HPLC analysis was acetonitrile. ACN stands for acetonitrile, methanol and water in the ratio of 10, 100 and 90. 10 part of acetonitrile, 100 part of methanol and 90 part of water. So, under this mobile phase and the flow rate for HPLC, the flow rate of mobile phase through the HPLC column was maintained at 0.5 ml per minute, and the injection volume for HPLC analysis was 20 microliters we have injected. The detection of our phytoconstituent in the HPLC was at 220 nanometer. The UV detector was set at 220 nanometers. The retention time for digoxin in the chromatogram was obtained at 12.9 minutes. It means that after injection, after the time of injection, it took 12.9 minutes for digoxin to show its peak in the chromatogram. So, at the time of injection you get a plain line, and after 12.9 minutes you suddenly get a peak, and that peak at 12.9 minutes shows the presence of digoxin in the sample. So, the sample for this analysis was sample preparation for this analysis was same as we have discussed in TLC analysis: 250 mg drug, 25 ml water, methanol, ultrasonication in ice bath for 1.5 hour, filtration, and the method which we have discussed in the TLC analysis. The sample standard was prepared for HPLC analysis by dissolving 1 mg per ml of mobile phase plus 2 ml of beta methyl digoxin as internal standard. Internal standard is something which makes the detection of your standard possible or easy for HPLC analysis or the UV detector. So, internal standard is added for both samples and for both standards also. So, this is the HPLC image of HPLC instrument. This is the solvent reservoir, this is the pump. Here we have the injection. This is the HPLC column, C18 column it is available C18 column, or in other analysis we can also use C8 column, and it is the detector. After moving through the column at different times, at 12.9 minute we can detect the digoxin through this detector. So, this is the systematic diagram, and the readings they are automatically fed to this computer. This is the this is the instrument which we can see. This is the sample holder. This is the solvent tray. This is the pump, and here we can place the column, or you can say the column incubator or column chamber, and this is the detector. Here we have the detector. So, this is the basic instrument, HPLC instrument, which we can use. This is the HPTLC instrument. Suppose we have developed the we have developed the TLC plate. We can analyze the TLC plate using this HPTLC technique. So, it is provided with the automatic sampler, automatic developing chamber, chromatogram immersion, TLC scanner, TLC visualizer, and the software for integration of all the facts. So, if we talk about the utilization of digoxin, we have seen that it is used for cardiac diseases, it is used for heart failure, it can used for treatment of arterial arrhythmias, fibrillation, arterial fibrillation or flutter. It has a narrow therapeutic index of 0.9 to 0.9 nanogram per ml, but it is not recommended in patients with right side heart failure. So, these are the specifications for digoxin, and we can use digoxin for treatment of heart failure and arterial fibrillation, to be used only when the diuretics and ACH acetylcholinesterase inhibitor they are failed to control the symptoms. So, this is the use of digoxin for medicinal use. And thank you so much in the next lecture we are going to cover the next topic, which is Taxol. Thank you so much.